wanted to tell you a little bit about bioMérieux and our role in diagnostic development. We develop and manufacture many of the IVD or in vitro diagnostic products that one would find in a clinical micro lab. These products, they range from the sample processing to ones that generate the final results that are sent to the clinician to help guide the most efficient antibiotic therapy. Diagnostics are key and critical in the fight to detect, control, and prevent antimicrobial resistance. In growth-based microbiology, which most of our diagnostics employ, methods are used to culture the microorganism that's causing the infection, such as with a blood culture system. Blood samples are taken from the patient that has symptoms of a bloodstream infection and cultured in broth bottles with an indicator system that can rapidly detect growth of the agent that's causing the infection. Once that agent is isolated, other tests are used to characterize the microorganism that's causing the infection. This includes identification, or telling the clinician its name, what bacterial or fungal species is causing the infection, and its susceptibility or resistance to various antibiotics. Today, in the clinical lab, there are two main methods for microbial ID, or identification. The first is one that uses biochemical reactions. It has very good resolution to identify a large number of species, but these tests can require anywhere from hours to a day to get the results. The second is a relatively new method that has revolutionized the workflow in the clinical microbiology lab. This newest ID method, it utilizes proteomics and has the highest bandwidth to identify many more species with the highest level of accuracy comparable to DNA sequencing. This works by analyzing a mass spectrum that is typically comprised of about 100 to 200 protein peaks. The proteins are highly conserved, and they're related to the DNA sequencing targets that are used for microbial identification and taxonomy. This platform is called MALDI-TOF MS. That stands for Matrix-Assisted Laser Desorption Ionization Time-of-Flight Mass Spectrometry. It's easy for me to say. While MALDI-TOF MS is not commonly used to characterize strain relatedness or determine that they belong to the same clone like you saw with the map of Candida auris, it's a highly accurate method. It gives species level identification, and it's within minutes of the sample preparation from the isolated colony. So these very rapid results allow the clinician to be able to refine the empirical therapy before antibiotic susceptibility test results are available, and have been shown to reduce patient mortality and reduce hospital stay. Antibiotic susceptibility test results, they vary in speed depending on the method. There's some rapid screening tests, like a beta-lactamase test or carbapenemase one. But for a full antibiogram, for the full spectrum of antibiotics of interest, methods that generate either quantitative results, which are called MICs, or minimum inhibitory concentrations, and their associated qualitative categories, S for susceptible, I for intermediate, or R for resistant. They require many hours to more than a day for the final results to get to the doctor. I prepared a handout that gives a brief description of the different types of antimicrobial susceptibility tests and identification methods. They're on the table in the hallway, as well as a few other handouts about antibiotics, stewardship, and blood culture, and one other. But if you need more information, you can find many more details on the internet. A quick Google search will bring you more information than you can handle. So thanks for your attention, and we'll move to the next.